Our next session is moderated by Dr. Jen Gomerman. Jen, welcome. Jen is the professor and chair of the Department of Immunology at the University of Toronto. Her research focuses on adaptive immunity, autoimmunity, gut immunity, and multiple sclerosis. She did her PhD in immunology at University of Toronto, postdoctoral fellowship at Harvard Medical School, and she holds a Canada Research Chair in tissue-specific immunity. She's been a massive contributor to the COVID Immunity Task Force and to COVARNet over the last couple of years. Thank you, Jen, for joining us, and I'll let you go. You're going to do something that's not easy to do. You're going to be moderating this panel virtually. So if you need any help, we'll do our best to help you. Your panelists are here.
Great. Thank you, Tim. Can everyone hear me okay? I'm actually working from home because I'm about to board a flight to Montreal so that I can be with you tomorrow, but I wasn't able to be there today. So I'm looking forward to continuing discussions in person tomorrow. I'd like to introduce the panelists for this discussion. With us here in person is Dr. Monica Strengert. Dr. Strengert is a virologist with a particular interest in host immune regulation after infection, vaccination, and pharmaceutical interventions. She currently leads the Adaptive Infectious Disease Diagnostics team within the Department of Epidemiology at the Helmholtz Center for Infection Research in Germany. Prior to her role at Helmholtz, Dr. Strengert contributed as lead biologist and program leader in GlaxoSmithKline's refractory respiratory inflammation discovery platform unit to developing novel medicines to prevent pathogen-driven COPD exacerbations. So I think what I'll do is I'll introduce speakers one at a time prior to giving their presentations. So I would ask Dr. Strengert to please go ahead with their presentation, and then I'll introduce the next speaker thereafter, if that makes sense.
The slides in front. So yeah, hi. Thanks for the introduction and the possibility to present. Can I actually also see this? Yeah. And the opportunity to present the essays we've developed in the last 4 years. In my talk, with a particularly focus on differentiating vaccination from infection-induced responses. As a brief recap, technology-wise, we use Luminex bead-based technology because the technology, by using a distinct color-coded bead, is extremely flexible, time and resource efficient because it allows the analysis of up to 500 parameters in a single measurement within one sample. When it comes to platform capacity, it is compatible with the standard laboratory equipment. However, with appropriate liquid handling infrastructure and automation, you can also process up to 1,500 samples a day, as it's happening right now at our collaborations at the NMI. I have been also asked to comment on assay development. In our hands, both during screening and development, we follow FDA immunoassay guidelines. We apply algorithm-based QC samples in order to reduce the amount of controls needed when we do multi-pathogen analysis per plate. And if possible, we also try to use international standards for quantification to make our assays comparable to other data. When it comes to biomaterial, generally we adjust our assays to serum and plasma of human and also other mammalian species. But for SARS-2, we also implemented saliva. As of course, it's a mucosal pathogen, so information about the immunity at the primary site of infections is very relevant. And as Helen has stated, we also try to use dried blood spots because the use of that kind of material has implications for pandemic preparedness and for cost effectiveness when participants can do home sampling. The pathogens and diseases we're interested in here are listed. Most of the assays are established and some of them are in development, and I'm today going to present some data on hepatitis A, measles, mumps, rubella, varicella, and of course, as everyone was also busy in the last years, we also developed assays towards SARS-CoV-2 and the common cold coronaviruses. As said, as we all were busy in the last 4 years, with SARS-2, and there we developed 2 assays. Multi-CoV measures presence of antibodies and antibody binding, whereas the second assay is a virus and cell-free surrogate neutralization assay, which measures functional capacity of antibodies, mainly to neutralize SARS-2 as a target. We have, what has been particularly useful using the multiplex platform was that we could basically fast incorporate the genomic evolution of SARS-CoV-2 by including current and emerging variants of concern, the globally dominating one like the multiple Omicrons, but also more locally spread variants like the mu variants. And with these 2 assays, we have by now screened more than 100,000 samples, which resulted in more than 25 publications. And we covered a vast array of studies ranging from seroprevalence to preclinical vaccine development, disease and vaccination response study. And the bullet point on the last one, the pre-existing and cross-immunity question, that was actually what kick-started our assay development early in 2020, because back then there was the hypothesis that pre-existing antibodies towards the common cold viruses could impact SARS-CoV-2 infection susceptibility. And in that respect, we have by now conducted multiple studies in the general population, in indigenous and urban children from Colombia, in household transmission settings, and also among severely ill individuals. But in contrast to T-cell-driven pre-existing immunity, in our hands, the B-cell-driven immunity has remained controversial, as observed by many others. And we believe this is, again, due to different study design which impact the conclusions. What this slide also illustrates is, however, that in the beginning everyone was focused on infection responses, and that interest also shifted in our work with the induction of multiple vaccines in late 2020. And as already stated yesterday, for SARS-2, that distinction is fairly straightforward by the presence of nucleocapsid antibodies only in infected individuals. And then once that distinction is made, we then moved on with our assays for a deep profiling of vaccination responses in the general population in Germany. And here we saw samples from the MUSPAT study, which is a 1-year study in Germany where we went to 8 different locations and sampled more than 40,000 individuals. And as others, we found that, of course, messenger RNA and mix and match vaccinations induced higher levels of antibody compared to heterologous vaccination, compared to vector-based vaccinations. However, what we were also able to show is then we went into a profiling of spike-specific antigens, is that the messenger RNA and mix and match vaccinations include more antibodies with a neutralizing capacity, whereas the vector-based one induced more S2 specific antibodies. And we believe this is an interesting molecular link towards the different clinical efficiencies we see in these vaccines. We applied our assays not only in the general population, but we were also able to profile neutralization responses in at-risk population for severe COVID. In our hands, that was a dialysis cohort that we basically followed from their first vaccination with Pfizer to now 3 doses of Pfizer, 1 dose Moderna, and now they got a bivalent vaccine. And what was particularly nice in using our setup, we could, every time we sampled our cohort, we could immediately show how well their vaccination status was working towards the novel dominating or novel appearing variants. At the same time, then some of the samplings we did, I also performed a pseudotype study, and just bit of lab-based background here. The pseudotype study was limited to 5 variants of concern, and I had to argue really hard to get the sample numbers close to 100 with our collaborators, whereas in our multiplex study, we would measure 100 samples on one plate in a day and not limiting the RBD's variants of concern that we could use, again highlighting the potential a multiplex system has. So I only have 2 minutes left. So, as I said, it's very straightforward to distinguish infection and vaccination in SARS-2 with nucleocapsid antibodies. It's also well accepted for hepatitis B virus with HBs antibodies and HBC antibodies. What we have been also able to show in the other assays we developed in the last year is that, now my slide is stuck, no. It's working. We have been also able to show that differentiation for hepatitis A virus. And there it works basically by the presence of 3C and 2AB specific antibodies, which are only raised if hepatitis A is actively replicating in infected individuals. And with the presence of these antibodies, we are now able to distinguish infected from vaccinated individuals with close to 90% sensitivity and specificity. And we're currently applying that assay in its first use case and real-world testing in a study we conducted in Bogota to basically assess the Colombian hepatitis A single-dose vaccine policy in children and if it's really efficient. And then the last assay that I'm going to present, it's an MMRV serology assay, which has 3 aims. The first aim is, in case of measles, to provide a sensitive and specific assay to accurately classify the serostatus, because currently there's an absence of a gold standard assay. And we perform that goal by a broad antigen profiling with multiple peptide and protein antigens as shown here on the bottom left. And then the second aim of this assay is by expanding the antigen panels to MRV to basically monitor the entire commonly used childhood vaccine as added value. And then, as Bill Moss has pointed out yesterday, the holy grail of measles serology, this is the major aim of the assay, that we have to differentiate infected and vaccinated individuals because you can provide evident information for measles eradication. And that was also one of the reasons why we performed the broad antigen profiling. And by now we have been able to demonstrate with the markers that are shown on the bottom left by a combinatorial analysis that with over 90% sensitivity and specificity, we can distinguish infected from vaccinated individuals. Now, this has been performed in a rather small training cohort with 150 individuals, and we're currently trying to expand it to a much larger end to see if it holds in that context too. With that, I'm done, and I hope I have been able to demonstrate that multiplex immunoassays can provide precision immunology estimates to seroprevalence study, and of course, listed below, acknowledging all our partners, our funding agencies, and particularly participants of our study who made our work possible.
Okay, thank you, Dr. Strengert. I was very pleased to see you're also looking at saliva, so we'll have to talk tomorrow. Our next speaker is joining us online, Dr. Alex Dulovic. He is a group leader for multiplex immunoassays at the Natural and Medical Sciences Institute at the University of Tübingen in Germany. Following his PhD on molecular tool development in parasitic worms at the Max Planck Institute for Biology, he joined the NMI in 2020 as a postdoctoral fellow and project leader. Now leading his own group, his research focuses on the development of multiplex immunoassays for respiratory, emerging, and climate change-influenced pathogens, with a particular emphasis on differentiating between infection and vaccine-derived immune responses. During the SARS-CoV-2 pandemic, Dr. Dulovic contributed to immunological studies of SARS-CoV-2 and continues to investigate biomarkers of post-COVID as well as the effects of NPIs on the immune status towards other pathogens. Looking forward to your talk.
Perfect. Thank you very much for the introduction. Can everyone hear me and see my screen? That would probably be a good start. Good. So yeah, thank you very much for the introduction. I will kind of pick up really where Monica just left off, as our two groups and departments work really closely together within the field of serology. But where she has given kind of more of this overview on our vaccination versus infection, and in particular, this kind of precision immunology stuff that we do, I will kind of now really focus on the work that we do more in a public health context. So fundamentally, obviously, when we talk about serological assays, yeah, we're talking about determining seroprevalence, protective titer, because we want to investigate some form of the spread or dynamics or susceptibility. But what I already talk about today is kind of coming from the assay development side. Well, how can we develop assays to identify reinfections, for example, within a population? If we have already 100% seroprevalence, obviously, their finding presence of an antibody is pretty much useless. And then particularly in this kind of preparedness direction, particularly some of the work we do now on tracking pathogens, and then also the rapid development in response to outbreaks. And when I was putting these slides together a week ago, I thought everyone would probably have had enough by now of the various SARS-CoV-2 related work. So I thought I'd pick some pathogens that were less likely to be represented, like RSV, tick-borne pathogens, and Mpox. But as I discovered yesterday during the first scientific session, it seems like there are several others here with the same idea. So to begin with, on really identifying these reinfections, for RSV, the entire population, broadly speaking, is seropositive. It's really only very young children who have somehow managed to avoid it during their first winter season, and who no longer have maternal antibodies that are negative. And I would just draw your attention here to this kind of rainbow-colored graph at the bottom on the left-hand side. And this is the post-F antibody titer for RSV. Post-F and pre-F represent about 90% of the antibodies for RSV. And I've split out the ages for kids under 6 into single years, but otherwise they're grouped together. And you can see it's just an absolute mess, really. There's no clear patterns of anything. Everyone is highly individualistic based upon their previous exposures, how active their own immune systems are, how long it's been since they've had their last infection. So this kind of gave us an idea that when we were developing this assay, that firstly, it was not going to be suitable to use random sampling. We were going to have to use longitudinal sampling. And that secondly, we were going to have to really develop an assay that very carefully, quantitatively tracked the actual differences and changes in titer in response to infection. And so this represented quite a technical challenge, because normally if you're developing an assay, you're just going to use positive samples versus negative. Well, here we had to firstly go to clinical trials, and particularly into challenge studies, and get samples from people who had been infected versus those who had been given a placebo. And then through profiling, we could really identify, okay, there are actually defined ratios that are highly indicative of reinfections or active infections having occurred. And we could also then, through the placebo samples and through these negatives, really gain an idea of actually, okay, what does decay look like? On the other hand, how fast are people seroreverting naturally towards RSV? And the second part was then a very strong technical challenge. If you are, as Monica said, when we develop assays, we do this to FDA bioanalytical guidelines. And for intra-assay variants, this is currently listed at about 15%. If you buy a kit off the shelf from a commercial manufacturer, this is roughly the variance that you will receive from it. And what we found is, obviously, if we're classifying a change or an infection as being a change in 15, 20% in the antibody titer, obviously we have to massively improve the variance. And this was really intensive work actually done by the PhD student here, Patrick Matzau. The work has now been published last week in Infection. And he went really deep into the actual chemistry, into the development of these multiplex assays. How can we physically bind the antigens to the beads? How can we optimize every step? And this had twofold benefits. Firstly, this assay actually runs with about a 4% CV. Some antigens were even under the 2% margin. And secondly, it actually introduced a series of antibodies that had been previously neglected within literature. So G protein antibodies for RSV, these are what really define RSV into actual subtypes. And everyone previously had neglected them or rejected them in assay development because, simply put, they didn't couple to beads very well, they didn't produce consistent results. And we found that when you really optimized it, actually the Gs were perhaps the most informative of all, because they respond very differently with actually whether it's been an RSV type A or an RSV type B infection. So particularly in younger adults and children, but also in the general population, you can have between 50 and 90% of your classified positive samples to be able to then subclassify them and say, actually, this person had an RSV subtype A or an RSV subtype B infection occurring. And so having developed this, we were then able to really go and introduce these into epidemiological studies. And we have one particular study which is still ongoing, where the same group or population of individuals have now been sampled 3 years successively, and they will be sampled for another 2 years. And so we could really track how the pathogen was spreading within the population on a year-by-year basis. And when we put this together into some modeling data, you can see here on the left-hand side, this is from the RKI, this is classical sentinel syndromic surveillance data, PCRs effectively. And here they always heavily report that in these purple and this red group, this is children under the age of 4, they are experiencing, or they are having the most amount of positive tests, which is logical. Children are the ones who get PCR tested when they show up at hospitals. But in the population, actually in the 2020-22 or 21-22 season, it was household transmission. It wasn't just the kids, their parents were also experiencing RSV infections. And then really in the following year, 22-23, when NPI measures from COVID were listed, we started having more spreading. Not only was the magnitude of the infections much larger, but also we had what we've now termed the grandparent effect. So effectively, the majority of the infections were actually occurring within individuals 50s, 60s, 70s-year-old, who would not be represented in PCRs because they simply put would not be tested normally. So this is kind of a nice example of how we could develop an assay and now already get some actual usable public health and modeling information from it. And obviously we're now following this for another 2 years. And we're also trying to develop an assay for flu to do something similar. I hope I don't have a clock, so I'm not entirely sure how I am for time, but if I have time, I would just go then briefly onto how we are for some of our other pathogens. And when I talk about pathogens of climate change, for me at least, this is tick-borne pathogens. You can see here on the left-hand side, ticks are prevalent absolutely everywhere within Central Europe, particularly in Germany. We have a suspected rate of about 0.03% of the population getting infected every year with Lyme disease. But of most concern, we are starting to see tick species that are not normally found within Central, Northern Europe, traditionally found in, say, Northern Africa, Southern Europe, Mediterranean, warmer climates, starting to make their way north. And this is, for an example here, Hyeloma marginatum on the right-hand side. This is a known vector for CCHF. So this is now a pathogen that we are starting. We've had the first couple of cases occurring within Northern Europe, and it is starting to spread further north. And so in the context of tick diseases, we actually a few years ago developed an assay for this, Borrelia multiplex. And the reason for this was purely so we could actually investigate Lyme disease spread in a seroprevalence context, because classically you have a two-step diagnostics from the serological side and ELISA on a Western blot, which means it's not really suitable for epidemiology. However, here, what we found is by using the IntelliFlex system from Luminex that allows dual classification or dual measurements of IgG and IgM at the same time, we could build an assay that was multi-analyte, so IgG-specific antigens, IgM-specific antigens, multi-species. We could ensure that all of the species of Borrelia that were present in Europe were being covered. And this multi-isotype together meant that we have a one-step assay that comes out with about 95, 93% spec. And we actually think, if anything, this is an underestimation because we always had to use as our control this so-called gold standard of the very poor two-step testing currently. And we did put this into epidemiology. I'm not going to go into this in too much detail, but we saw that there were some differences geographically, some age differences, some gender differences. But I've more highlighted this because this assay is now something that we are going to use repeatedly in the coming years, because Lyme disease is something of a warning sign. If we start seeing that seroprevalence is massively increasing, particularly in coming years, that it's occurring more commonly in younger individuals, this is a warning for us that these rarer diseases are also likely circulating. And the assay itself can also start as a basis point for us to expand out with this Luminex technology and add new antigens from these other tick-borne pathogens. Last point then really just on rapid development. So we obviously did this in the context of SARS-CoV-2, but we also did this in the context of the Mpox outbreak. So in 2022, we started with a very broad assay development. We took, I think it's 14 different antigens roughly from Mpox and vaccinia. And the only part I really want to draw your attention to here is actually this grouping on the left-hand side of these heat maps. And what was critical is that we saw very early on, within weeks basically of developing the assay, that critically the grouping was not occurring on the basis of the actual species, but instead it was based on the antigens themselves. So this gave us a very strong indication that the antibodies from historical smallpox vaccination, from other orthopox viruses were going to be cross-reactive towards mpox. And I know yesterday someone presented data where they said that E8L or E8 was the most informative for them within their assay. For some reason, actually in Europe, so other groups, not just ours, but also people at UKHSA, and I think also in Norway and the Netherlands, we all found that B2R for some reason was the most informative. This gave us an overall sensitivity specificity of 96, 97, 98 roughly. And we found that this actually, while the E8L was definitely one of the 3 or 4 best performing antigens, there was this small, I think it was about 4% increase in sensitivity we could gain with this other antigen. So we already actually had then done this work in August of 2022. So while the outbreak was still sort of wrapping up to a degree. And we could see here by putting our cutoff line and by putting in, I think it was about 1,300, 1,400 samples from the population, we could assess what titers were actually already present in the populations. These were samples collected in 2021 prior to this outbreak. And the critical part here is that obviously young kids, young adults, things like that, they're all clustering on the bottom side. But as soon as the smallpox vaccination comes in, everyone starts to come up to meet really this cutoff for positivity. What's really interesting though, and what we're now investigating in more detail is there is about 10% of these individuals who would be classified as positive who have no historic smallpox vaccination. This represents about 1% of your population overall. And this means that there are other unknown currently orthopox viruses circulating within Germany. And so we're really now kind of using this and actually going further with the epi analysis to really investigate and try and figure out what actually is circulating within this field, because obviously these pathogens are so closely related. And with that, I hope I've managed to give a very brief sort of whistle-stop tour of some of the work that we do. And I would thank all of the various funding organizations, partner institutes, in particular the HARDZD, that we have worked with, as well as yourselves for allowing me to present my work. Thank you.
Great, thank you very much. And so our final panelist is Dr. Emilia Liana Falcon. She is the director of the IRCM Post-COVID-19 Research Clinic and Microbiome and Mucosal Defense Research Unit at the Montreal Clinical Research Institute. Her research unit focuses on the study of the role of the microbiota in driving local and systemic immune responses in patients with specific primary immune deficiencies. She's also an assistant clinical professor at Université de Montréal, an infectious disease specialist at Centre Hospitalier de l'Université de Montréal, and holds a Canada Research Chair in the role of the microbiome in primary immunodeficiency. During the COVID-19 pandemic, Dr. Falcon established the IRCM Post-COVID-19 Research Clinic, the first of its kind in Quebec, and has since evaluated hundreds of patients with long COVID-19. Welcome.
Great. Thank you for that introduction, and thank you for the invitation to present today. So I have to say, I'm really impressed with the caliber of the presentation, the work presented during this conference. So as you can see, I'm not an epidemiologist. I'm not a specialist in public health, but I'm an ID doc that likes to play in the lab, very interested in identifying biomarkers. I worked a lot with rare diseases. Now with long COVID, it's a lot less rare. And so with that interest in biomarker discovery, I actually became the biomedical co-lead for the Long COVID Web, which is a pan-Canadian long COVID network led by Angela Chung. And I also chair the Canadian guideline team for the diagnosis of post-COVID conditions. So the question is, how did I wind up here today? So I was on a flight to DC to give a talk at NIH. And I noticed Tim, and I was like, he's important at CITF, and they gave me money. I should talk to him. So I stopped watching season 6 of Sex and the City, and I approached him, and I started telling him about all my mechanistic work. But really, what I wanted to do is find a way to truly integrate biomarker research and the evaluation of biomarkers in the clinic, like everywhere. And then he said, well, have you heard of Hemanet? And I was like, oh, this is amazing, blew my mind, and my to-do list got longer, and I'm here today. So right before the pandemic, I established my lab at IRCM, and we were focused on inborn errors of immunity, looking at how the microbiome drives severity and penetrance in these diseases, but also looking at how these diseases influence the function of the microbes. And there we have the study of in-host adaptation of bacteria. And then with the pandemic, we started studying long COVID, and we established this cohort with Biobank, which is a great infrastructure for interventional trials and looking at the impact of vaccination and reinfection in this context. And we got interested in looking at immune dysregulation, viral persistence, and the role of the microbiota. So the IPCO clinic, as we call it, or IRCM Post-COVID Research Clinic, was really the first clinic of its kind in the province because it really integrates a clinical evaluation done by myself in the context of a research protocol. So the clinical evaluation was extensive and standardized across the board. The visits are very long. But we also do extensive sampling and biobanking. So we banked saliva, whole blood for serum, plasma, PBMCs, urine and stool, of course. And we were able to just quickly go to my lab on the next floor and start studying mechanism. So patients could join our protocol or be seen in our clinic at any time in their post-COVID course, but the schedule is fixed at 1 month, 3 months, 6 months, 12 months, and 24 months. And then we added on visits after reinfection, so they're offered a visit at 3 months and 6 months post-reinfection, and then they're also offered a visit 2 to 4 weeks after any COVID vaccine. So the way our structure is was actually really convenient. So I don't get my 10,000 steps a day because my lab is really right above the clinic. And we really have a structure to do great mechanistic work. So from immunophenotyping to sequencing to proteomics and metabolomics in our core. But we also do a lot of culturomics, so isolation of very interesting candidate bacteria and their identification and their functional evaluation. And we do in vivo models, including in germ-free mice. And we like 2D, 3D preclinical models as well. So we work with organoids, but we're also trying to now incorporate organ-on-a-chip systems that are personalized. So in case you can't tell, I obviously have a bias towards the role of dysbiosis or the perturbation of the intestinal microbiota and what it does at the level of the intestinal barrier and how that then can drive long-term immune dysregulation, as you'll see in some of our data, and ultimately end organ damage. And this we've seen in the context of post-COVID conditions. So quick view on what our cohort looks like. So it's really small compared to the cohorts you work with, but in essence, it reflects what's been published in a lot of studies. So most of our participants were female, they were middle-aged, and they had a what we call non-severe acute COVID-19 infection. And what we really see when we did clustering analysis of all the symptoms that we queried for is that we really have these two clusters. One that's very driven by neurological symptoms. So this combined with fatigue, of course, includes trouble with memory, headaches, trouble with concentration, the famous brain fog, issues with sleep, and then sort of everything else. And so this neuro sort of cluster is really what has a lot of our cohort being affected for a very significantly and for a very long time, like in the measure of years. So the first thing we did is we looked at their microbiota. And what we found is the individuals in cluster 1 that were really neuro-driven, they had obviously less microbial diversity, but they also had a distinct microbiome signature with distinct function. And to just sort of see what the impact, and if we could transfer this phenotype just by the microbiota, well, we colonized germ-free mice. And so what we found, and these are not infected mice, okay? What we found is the mice that were colonized with the stool from patients who had the cluster 1 neurological, severe symptoms, they really performed more poorly on a variety of neurobehavioral mouse tests, including tests for anxiety and memory. So we wanted, I'm not showing this data, and this is really a quick high-level sort of view of our mechanistic data, but we also looked at neuroinflammation, and they have neuroinflammation, and we really wanted to make this sort of connection between dysbiosis and end organ damage in long COVID. So we started by looking at the barrier, and zonulin's a marker you could look at in the plasma, and when you have increased levels of zonulin, you have more intestinal barrier compromise, and we saw that if there was more severe disease, there was more evidence of intestinal barrier compromise, and this was directly correlated with increased translocation of microbial byproducts, such as LPS binding protein, beta-D-glucan, for example. So then we wanted to make the link between intestinal barrier dysfunction and immune dysregulation. So we I have a group of individuals that did research in HIV. They're very interested in B cells, and they said, well, you really need to look at BAF, so B cell activating factor. And we found that levels of BAF were also positively correlated with markers of intestinal barrier compromise. And this was also associated with changes in our B cell frequencies, including increased frequencies of these B cells, which we call precursor marginal zone B cells, that predispose towards autoimmunity. And we see this at their initial visit, but we see this even more pronounced, this dysregulation at 12 months post-COVID, and now we've looked even at 24 months, and we still see it in those who are symptomatic. So, of course, we then wanted to see, well, do we see more autoimmunity? We know there's studies that show that there's increased autoimmunity in long COVID, and we first started using a broad panel in collaboration with Aaron Ring at Yale. It's called REAP, and here he has like 6,500 protein targets. And we really saw like our patients who had this immune dysregulation seem to have more of these autoantibodies. And we're now validating this with the more clinically available autoantibodies. And these levels of these autoantibodies directly correlate again with these precursor MZB cells. So the major outcomes of this work, and I said this is a very quick aspect, and I didn't nearly get into the microbiology of all this, is that we were managed to identify a new biomarker panel to help diagnose severe long COVID and to track it over time, so to see how is response to therapy and so on. We've also developed a new method to detect microbiota-derived particles in plasma. So here, what we're talking about is what I call a fingerprint of the intestinal barrier dysfunction, has demonstrated by bits of microbes that are circulating in the plasma and that are unique according to the disease severity. So there is some similarity with cell-free DNA sequencing to look at microbes. We know this is this is up and coming, very hot in the cancer field. But here we're looking at we're developing a method to make this more adaptable to the clinic so that it's quicker based on a slightly different technology. And we've also identified a new treatment target for long COVID, for which we have a preclinical model. So, of course, the logical sequelae to me is to monitor for post-infectious conditions, harnessing a serosurveillance platform like HemaNet. And this, of course, would be with the integration of different types of biomarkers. As we know, they come in different shapes and sizes. We have molecular approach, a lot of immunoassays, a lot of the omics, especially metabolomics and proteomics. But we also should look at what comes from the host combined with what comes from the microbiota. And the interest in harnessing the surveillance or the follow-up of biomarkers to track non-communicable diseases, chronic diseases, we know is important. It's been raised yesterday in quite a few talks, and it's been done in lots of big cohort studies, lots in Canada, which I don't display there. I didn't want to, inadvertently leave anyone out. And to me, it's just a logical synergy that if you're going to look at chronic diseases, you're going to look at post-infectious conditions and how they can then lead to more of these end organ damages and chronic diseases. And the benefits for me are very similar, right? We would have now a biological estimate of the prevalence of different post-COVID conditions. We would then have the long-term follow-up now of what happens after known and emerging communicable diseases. And also, how do these post-infectious conditions impact aging and the incidence of other chronic illnesses? And then, of course, we would be identifying groups that are most at risk for certain conditions. This would lead to improved resource allocation and access to care. And new insights into intrinsic and extrinsic factors driving post-infectious and chronic conditions. And to come back to the discussion of yesterday of, do we sell this about public health surveillance or about research? To me, it really has to be both. So, a Public Health Agency of Canada has a wonderful platform to track chronic conditions. It's beautiful. It's very detailed and longitudinal. And I can totally visualize there a new panel looking at post-infectious conditions. Of course, also integrating various biomarker measurements. And my take-home message is, after giving you this very broad overview, is that long COVID really has been the poster child for post-infectious conditions. And I think there's so much more we have to discover about other infections because you don't know what you don't look for. And the thing is, these are post-infectious conditions are the new chronic diseases, and they may really alter our aging patterns. We already see that, the brain of someone who had COVID has aged like 20 years. I mean, this is scary stuff. And this will really have an impact on resource allocations. But of course, as we've realized with the literature, post-infectious conditions are clinically complex, they're heterogeneous, which is all the more reason why we need to measure biomarkers to support this diagnosis, monitor response to interventions, and even properly randomize people in interventional studies. And of course, the synergy between longitudinal serosurveillance and the study of these biomarkers, whether they be immunological or measures of end organ damage, would definitely lead to novel clinical and scientific insights. So this is my wonderful team, and big thanks to them and our funders and our patients, of course, who teach us a lot every day. Thank you.
Merci beaucoup, Dr. Falcon. And so now we will move into the panel portion of this session. Some of our objectives here today are to really delve into advanced scientific approaches supported by serosurveillance, like precision immunology and assay development, discuss applications and implications, and explore future research directions. So I'm going to open this up with some questions before we turn it over to general questions from the participants. And this is for any member on the panel who wants to weigh in. What advanced scientific approaches are being supported by serosurveillance, and what are their potential impacts? And I think everyone touched on this a little bit in their talk, but maybe if you could name the one thing that you think is really the most advanced scientific approach that could really make a difference and move the needle in serosurveillance, what do you think that would be? And feel free to just jump in.
Perhaps I start from online. I would personally say, regardless of the platform you use, I think multiplexing is the fairly obvious answer here. Whether this is a multi-analyte perspective, whether this is a multi-pathogen perspective, I think here is really where we have the opportunity, we have tools, we have various platforms where we can push this forward. And it's really now about actually putting in the effort, developing these assays, particularly standardizing them, because standardization of multiplex assays is a very tricky and a very time-intensive task. But this is where I think if there is some consensus and if there are multiple groups moving forward, that's where we can make the most benefits.
Anything else from the other two speakers?
Well, I would agree with that. I think that's really, really critical. But I would say that we have to start thinking also about improved technology to sequence microbial genomes in the plasma and traces of this. I think this will tell us a lot about where a person's been, where they're going, what's happening to their body.
And I think that kind of leads us into this other question of what future research directions should serosurveillance explore beyond? And do you see a role for AI or other technology platforms that maybe aren't quite there yet, but maybe we should be thinking about?
So I can say perhaps from personal experience, sorry if I've jumped in over anyone on the panel. We do some work now with AI-based work, really, and this is actually massively helping, particularly in investigation of biomarkers and really actually identifying unique targets that we would not think of, particularly if we're looking in the field of modifying some of our proteins to actually expose epitopes that would not be naturally available, but actually would bind in reality. This is where something where AI is making a big difference and a big jump forwards, particularly from taking this kind of top-down approach of, well, I've got a disease of interest, how do I find a marker that actually allows me to investigate it further?
Yeah, again, I mean, huge role for AI, but I think that one of the things is we have to be able to feed a lot of data into it. We would like to feed a lot of biomarker data into these sort of systems. And for that, well, it comes back to having infrastructure to generate this data and getting over all the consent hurdles that we talked about yesterday, which, you know, I think right now is our limitation and causes a big bottleneck.
Yes, I guess the correlate to that question are the ethical concerns around feeding metadata into these types of machine learning approaches. And yeah, I think that's definitely worth discussion. I have a question specifically for Dr. Falcon. Your talk reminded me of one of the neurological readouts that's used in my field in MS, which is serum neurofilament light chain. It's often used in trials, but it's at very low abundance, and so you have to use like a Simoa type approach to detect it. And so I'm just wondering, it's reflecting on your comment, we don't see what we don't look for. Are we missing potentially low abundance biomarkers that could prove quite powerful? And how do we deal with that moving forward from a discovery all the way to an application phase?
So first of all, Jen, I'm a big fan of you and your work. So I'm actually really happy you're moderating. You asked me this question, and I'm making lots of ties between what we're finding and connections with MS. And I think we have to take the biomarkers identified in MS and some of the stuff that we found and look in both patient populations and other instances of autoimmune conditions. So I think it's extremely important to improve our sensitivity of some of these markers. And that's sort of one of the issues, right, with some of the multiplex technologies, and maybe not necessarily in the context of serosurveillance, but certainly in the context of certain targets that I look for, is that when you go really broad, you lose a lot of sensitivity. You got to really play around with the different platforms. And I'm sure there's even better ways to get more precision, because sometimes it's just like in the microbiome. You know, we tend to sort of eliminate the ones that were low abundance, like we're thinking they're not important, but sometimes you just need a little bit of something to signal something that's going very wrong. So increasing sensitivity, top priority.
Anybody want to add to that?
So I was speaking with Thomas before, and he was saying that now everybody seems to be multiplex assays, and I think that's really great, because as Alex has pointed out, I think the potential lies in the information density that you can get from the measurement. And I still vividly recall early discussions that we had in the pandemic where we heard that our assays are not needed, not wanted, they will not work anyway. So coming these 4 years, I think the pandemic has incredibly helped to establish the technique, to show what it can do. And in our institute, we are now also trying to basically position the technique as a proper omics technique, together with genomics, metabolomics, and proteomics, to set our project up kind of like in a trinity way, and not just having, it's seroprevalence or presence of antibodies, but really to showcase what such an assay can deliver. And we have 2 projects where we kind of integrated it. One of them is the National Health Cohort of Germany, where we now do a feasibility study, not unlike the UK Biobank, where we have 5,000 participants. We have urine, plasma, stool, and blood, and we use serolomics, genomics, proteomics, and metabolomics to just showcase the feasibility in a population-based setting.
Very interesting.
So just to follow up on that, do you think that composite readouts might be one of the futures in moving forward? So, you know, maybe like a stacked type readout between a protein, an antibody, a cytokine, for example. What are your thoughts on that?
So with my pharma background, again, we rarely ran singleplex analysis, but all the time multiplex. So I think you need to really carefully look at the data and look where the biology leads you. And then, of course, that's also the next issue that we very frequently encounter. We're really lacking the bioinformatic capacity, and in particularly trained individuals who understand the programming, but at the same time understand the biology. Because also in my pharma background, sometimes our statisticians were hard at work, and they were so excited about COPD phenotypes, and they were actually regulating just the COPD phenotype, and they didn't understand the biology. Like, you don't want more mucus in a COPD patient, but this is what the compound was doing, and they didn't understand the background. So in the end, I think it's a combination of assay development, the proper data scientists, and also people who understand the biology to get the best outcome.
Yeah, I agree. I think these different types of approaches really need to work together to make bigger leaps forward. We're going to open it up to the floor now for questions. Someone over who's live is going to have to invite people.
Yeah, I'll jump up, Jen. We have our first, Lori, go ahead, please.
Great, thank you. Hi, I'm Lori Beach. I'm a clinical biochemist. So my question to the panel is, as we're talking about multiplex assays and how awesome they are, I'm wondering if you can comment on the translatability to the clinical laboratory, and really just to make sure that as we discover or find new biomarkers that we're really excited about that need really, really tight sensitivity, for example, that maybe we find could be amazing to move into clinical practice through HumanNet, through some of our sero studies. How do we ensure access for all Canadians for what is potentially a really specialized test? So just curious about how we can move multiplex assays into routine clinical use. Thanks.
So I hope that for multiplex assays, the same trend will apply then for gene expert technology, which is now also very accepted in third world countries and which is usable there. So I've been working in a hospital in Haiti, and they have managed to do these tests, and they were pretty good in destroying a lot of equipment. So I think I do hope that we're going to have the same push for the technology, like PCR was not common 30 years ago, Cepheid was not common, it is now getting more common, but I think that needs to be also like a common push of everyone.
Okay.
I would perhaps just add, sorry.
Go ahead, please.
I would just perhaps add to this very quickly. During the COVID pandemic, we've worked with a fair number of public health laboratories in other countries, different regions of the world, and largely the technology is there. They all have, analyzers capable of these multiplex measurements. It's just that fundamentally there is a staff issue to a degree, and fundamentally there is a usability issue. A lot of these assays we're talking about are not off the shelf ones. They are specialized. They're coming from academic groups, and it's perhaps more of a case of we need to start getting better transitions from the academic side out into industry, because obviously they can only really use stuff that is properly IVD certified.
Yeah, and I think this addresses an important question around global equity and how are we going to make sure that some of these assays translate into other settings where disparities have existed. Another question from the floor.
Yeah, please, Steve.
Hi, my question is to Monica. So you kind of briefly described an anti-hepatitis A virus assay, and we know canonically that protection is probably, between 10 and 20 IU per liter. Is your system able to quantify those levels?
So in that assay, we haven't used the international standard yet. That's also strongly dependent on which sample cohort we use for evaluating or setting up the assay. But we can, once the assay is done in that respect, we can speak again.
So I'll tell you my thing here. So as a blood operator, we send stuff off. All our plasma from whole blood donations and source plasma gets sent to 2 fractionators who test every single donation for hepatitis A and parvovirus B19. And so when I started at CBS, I was getting these spreadsheets of aggregate Hep A PCR data from 2015 to 2022, which represent 2.7 million donations. And everything was tested for HAV by PCR. And when we did the NAT positivity rates, they were 0.001% before the pandemic, and then after the pandemic, they dropped a whole log for positivity for NAT. Well, Hep A is not endemic in Canada, vaccines out there, but I don't know what the reviewer said, what's your positivity rate for serology? I'm like, I don't know, but it's changing. So if I had a quant assay and someone could do it, well, then I could figure out what the population level protection was, especially with the population changing. People coming from different places, they've been more exposed. But we don't know actually from public health what our seroprotection level is right now. Now, the thing is every donation I get also has an EDTA tube attached. We used to use those for COVID, but now I could easily just take them and run a population level protection level. But you can quantify, right?
We should be able to
Okay,
quantify
that's a good
once
idea.
the assay is established.
So it's more of a question, me trying to figure out how to advance my research career here, but it'll work.
To be determined, I think. We have another couple of questions here, Jen.
Hi, thanks for your presentations. This one's for Dr. Falcone. Thanks a lot for your great data. It was really good. And just kind of following up on the point of like the potential for long COVID being the poster child for persisting chronic disease after infection. So I would just sort of encourage also Hemanet too, just to include that chronic viral infections, of course, HIV and hepatitis C have had a long-standing interest in understanding now what when you have viral control or viral cure, and they have a significant presence of chronic disease. So I think your initiative for including this as a tab on PHAC is money, but should also include those other conditions. And I'm sure you know this, of course, speaking to an ID. Yeah.
No, you're absolutely right. And in fact, like I said, you know, we look to HIV actually for how to approach, you know, the study of the immune dysregulation. And, you know, I myself once upon a time looked at coronary artery disease in HIV. So I should have said that first. You're right. I think we're biased by the whole, you know, obviously having just come out of the pandemic and it's a respiratory virus. But much like in HIV, we're looking for that reservoir. So indeed, your point is excellent.
Great. And Jen, we have one more here.
Thank you to all the speakers for really wonderful presentations. I wanted to explore the other side of accessibility and sort of how many people can benefit from these essays. And I kind of want to maybe argue that not every site in the world really needs access to an assay that's designed for screening rather than for acute diagnostics. You know, if you have a patient presenting with TB or MRSA, you want to be able to diagnose them up front, as they present to the hospital, because the intervention is going to be immediate. For screening, perhaps, you'd have a bit more of a like laxity with how much you can, how much time can elapse between the sample collection and diagnosis. So that gives you an opportunity to transport your sample from the point of collection to a more centralized facility that actually can offer the testing. But of course, to be able to do that, you do need to have appropriate, transport routes, and you do need to have samples that are amenable, to this transfer, that are stable. So from your perspectives, the current samples that you're utilizing, the biomarkers that you're looking at, how stable are they? And, would there be opportunities to actually engage in building transport networks rather than localized testing facilities?
Any answers to that, Dr. Dulovic?
So on stability, I mean, I think perhaps something we always underestimate with serology is that we need to be better at validation to a degree, but particularly validation across multiple populations. We know for a fact that our assays have worked differently and have had different, sensitivity specificities within populations in Europe, for example, let alone before we started testing them in other countries. In terms of stability of them, actually, we have shipped this kit to 4 different continents. Successfully. It's been established in several different labs, the SARS-CoV-2 multiplex that is at least. And so actually that part seems to work well. What I would say is perhaps more the issue, and to a degree on the sample stability, particularly if you're going out into areas where the infrastructure is not available and need to bring samples back. There is a critical issue to a degree of converting these serum-based assays to be working with dry blood spots. Sensitivity specificity does reduce significantly for DBS compared to, say, serum or plasma. And I think it's more perhaps a question of parasites themselves or what exactly you are looking for, whether you can move past these issues. Obviously, for the RSV assay we worked on, that will not be possible with DBS, realistically, because it is so dependent on the sensitivity of the assay. However, for basic prevalence, if you've got Lyme disease or something like that, that would be something that would be applicable to work with DBS technology. So I think there's a degree of trying to strike a balance between where do we really need to keep with whole blood samples, serum, particularly cold storage routes in areas where this is not possible, versus where can we work with sample material that is, transportable at room temperature and things like that. And particularly, can we then develop our assays further to work with biomaterials that are more stable and don't require this.
Great, thank you. I think we have run out of time, unfortunately, so the remaining question, maybe you can find each other during lunch. I want to thank all three speakers for giving really inspiring presentations, and I'm looking forward to seeing all of you tomorrow at 8 o'clock in the morning, and sorry I couldn't be there in person today, but enjoy the rest of your day. Thank you.
Thanks so much, Jen.